Okay, today we're talking about errors that can happen outside of DNA replication, meiosis, or just by living. This is our mutations, okay, and they're caused by things called mutagens, chemicals, lights. Any agent that causes a genetic mutation is a mutagen. So we're going to talk about these damages in the DNA. Okay, and so mutagens are able to chemically change or disrupt the bonds between those bases, and then they're going to replace a nitrogenous base in the DNA sequence. So it's causing a change in the sequence of the DNA. So here again is an overview of the things that can cause these. These are mutagens, radiation from the sun or X-rays. That's why it's important to wear sun protection, and also to not take a lot of X-rays. A lot of times if you go to an X-ray, get an X-ray done, they'll put like a little lead protection thing over you, like an apron, and that is to protect the rest of your body from that radiation. Chemicals, so like carcinogens, preservatives, and different things in cosmetics and cleaning products can cause mutations, as well as some bacteria and viruses. Not all, but there are some that can. Okay, and then these are things that cause errors, but when the error is not able to be fixed, they're mutations. So a mutagen can cause an error and then it be fixed by the mechanisms we have in our cells. But when they're not fixed, it's a mutation. Okay, so we can also get mutations from meiosis, and this is dealing with the number of chromosomes that we would get. And we've talked about this some, but it could result in something called triploidy, where we have 3 copies of a chromosome. And it exists, and we can have sterility in some animals with triploidy. We can also have polyploidy, same idea. These words ending in ploidy make us think of like haploid and diploid. So triploid would have an extra copy. Polyploidy would have even more copies. So it's multiple sets of homologous chromosomes. Okay, examples of this are we've talked about Down syndrome having an extra copy of a chromosome, Turner syndrome. So there's some examples of this. Trisomy 21 is the same as Down syndrome. And so we've talked about this. What would cause this? Do you guys remember what it's called? It's called non-disjunction. So that's separating of our chromosomes, and meiosis doesn't happen properly. So these are also forms of mutations. Okay, so here's showing non-disjunction, where normally we would have our cell, and then after our rounds of meiosis, we end up with half the number of cells. So a diploid cell, or starting with a diploid cell, ending with a haploid cell. But if they don't separate correctly, we could end up with too many or not enough. So that would be non-disjunction. Okay, so mutations, the effect of them can be beneficial, detrimental, or neutral depending on the phenotype of and on the environment that it's in. So a particular phenotype can be beneficial under environmental conditions and detrimental under others. This is getting at kind of our idea of natural selection, that in different environments, something could be beneficial, and in a different environment, it could no longer be beneficial. Okay, but these mutations are affecting the phenotype, and depending on the environment, depends on if they are helpful or not. Okay, mutations also are causing a source of variation. So variation is helpful in a population. Shown here, we have lots of different giraffes. Okay, so this is a basis for natural selection, and we're going to talk more about this in our next unit, but just keep in mind that mutations can cause variation in a population. Okay, we can also have mutations being passed to other individuals in a population by something called horizontal transfer. Okay, and this is mostly done in prokaryotes. I can't think of a eukaryotic example off the top of my head, but it's primarily in prokaryotes. So this is where we are taking DNA and transferring it from one individual to another. So it's making it so that a mutation can be passed in the population very easily. Examples of this are something called transformation, which we will actually do in a lab. And this is where we're going to have DNA usually in the form of a plasmid, which is just a small circular piece of DNA that is then taken up by a bacteria or prokaryote. We can also have transduction, similar idea, but the DNA is coming from a viral infection. I guess this would be an example of how we can have horizontal transfer in eukaryotes if there is a viral infection that causes a DNA transfer. So that would be the only one. And then conjugation is another type where we would have DNA that's transferred directly from one organism to another. Okay, let's look at an example of this. An example of this is in bananas, in the Cavendish bananas. So Cavendish bananas are a triploidy. They're triploids. So they're actually sterile, and so they can't reproduce or produce seeds. And so it's because we crossed a diploid individual and a diploid gamete and a haploid gamete. So this is causing a triploid. Okay, so this is what a Cavendish banana. It's the banana we're typically used to, but naturally bananas would occur like this. And so the benefit of doing this is because they don't have seeds. And so then we don't have the seeds in the bananas. Okay, another example of this would be polyploidy, is in strawberries and other produce. Have you ever seen strawberries or different produce that look kind of funky with extra little parts to them like this? This is because they have multiple sets of chromosomes. So our wild type, that's what WT stands for, is just a regular diploid individual. And then when we have a polyploidy, we have it being larger. It usually has increased vigor and size in this case for strawberries. Okay, so mutations, these are again those genetic changes that can then be passed down to future generations or offsprings. Okay, and they can change what proteins are made because we're changing that DNA sequence. Or they could change parts of the DNA that are used to signal for replication enzymes or for the gene expression. Okay, so we have a couple different types of mutations. One is a point mutation. It's also called a substitution mutation, where we are changing one nucleotide for another. So in this sequence, we had an A here that's changed to a C. Okay, so one base replaced by another one. In these, only one I shouldn't have that picture up yet, but in these, only one amino acid is changed or affected because we're only changing that particular codon. Okay, we can also have types of mutations called frame shift mutations. These are from are going to cause the amino acid sequence to change, and they're because we are either inserting or deleting a base from the DNA sequence. So in this example, we are inserting an extra adenine into the sequence, and it causes the codons to shift so that now not only is this amino acid changed, it also affects every other amino acid downstream. So you can kind of imagine that a frame shift mutation is probably going to be more detrimental. Yep, they're going to change all those amino acids following the mutation. Okay, so if we can kind of think about this or symbolize it as a sentence, and each of our codons are 3 nucleotides long, just like this sentence is made of only 3 letter words. Okay, so what if we delete the letter C in cat? Okay, but we're still going to have every word be 3 letters long. So if we did that, it would be the fat, and then it goes into kind of gibberish after that because the C is deleted, but we're keeping our sets of 3 afterward. This is how we would have a frame shift mutation affect the rest of the sequence. What if we were to add an A after the word cat? We would have a similar type of result where, again, if we add an A, it will then shift our, have a frame shift because everything afterward is going to be kind of gibberish. What if we replace the letter C, though, with the letter H? So this would be a substitution or a point mutation. And in that case, it would change our sentence to the fat hat ate the wee rat. So it does change something about our sequence and therefore our protein, but it's only affecting that one amino acid or word in this case. Okay, so I want you to think about which one of these in this example are point or substitution mutations, which are deletion, which are insertion, and which are frame shift. So you can pause the video and try to identify them. I've kind of mentioned some of them. Okay, you should have identified them from these examples up here. So a point or substitution change would be this last one. A deletion change is this first one. Insertion, the second one, and the frame shift are these 2. Okay. We can also have different outcomes that we have from a mutation, and we call these either missense, nonsense, or silent. So a missense mutation, this is when the base was changed so that the codon is into a different amino acid. Okay, so that was like this example where we changed it to be a different amino acid. A nonsense mutation, this is when we change the sequence and it turns a codon into a stop codon. So this would like be if we replaced something with a period and it just stops it early. So this would end up with a shortened version of the protein. And then we can also have a silent mutation, and this is when it's going to actually not change the amino acid sequence. So you might remember when we looked at our charts for looking at codons that there are some that code for the same amino acid. So in this case, GAA and GAG both code for the same amino acid. So this mutation would not affect anything, so it would be a silent mutation. Okay, not all mutations are bad or even affect the organism. Okay, so this is looking at our idea of evolution again, and genetic variation is only possible because of mutations. So some mutations are beneficial. It gives us more variety, like shown here. These mutations are going to allow organisms to adapt and give rise to other organisms. Okay, mutations are why we are able to have people able to digest lactose. So in the past, just like other organisms, our young drink milk, but over time we kind of lose that enzyme to break down lactose. But because of a mutation, we now have people who are able to be not lactose intolerant and drink milk later on. And like we mentioned before, some mutations do not change the cell's function or ability to create proteins. So it could be that it is a mutation in one of our introns that's going to be taken out. They don't matter as much. Yep. It could matter though. Remember when we talked about our alternative splicing, so it could be incorporated into a different protein. Or it may code for the exact same amino acid like I mentioned in those silent mutations. These examples of codons that multiples code for the same amino acid, it's usually the last nucleotide that's being changed. This is called a wobble.